Hello everyone. Today in this animation, we will see the mechanism or steps involved in the process of phagocytosis. So first, before going to the animation, let's just talk about some basics of phagocytosis, mainly the mechanism of killing of bacteria by macrophages through the process of phagocytosis present on their surface. Phagocytosis is a fundamental process for the ingestion and elimination of microbial pathogens, foreign substances, and apoptotic cell particles larger than 0.5 micrometers in diameter. The professional phagocytes, like macrophages, express several phagocytic receptors like opsonin that activate signaling pathways resulting in phagocytosis. Now let's discuss the phases of phagocytosis. The first step is detection of the particle and its opsonization, followed by the second step of its engulfment and internalization process. The third step is formation of a specialized vacuole called phagosome. In the fourth step, phagolysosome is formed. Lastly, in the fifth step, the phagolysosomal enzymes degrade ingested particle. Now let's understand the animation. As previously mentioned, the first step is opsonization. The bacteria is moving freely in the bloodstream. The opsonin, which includes Fc fragments of immunoglobulin G and C3b, coat the bacteria and present it to the macrophage by binding to Fc and C3b receptors present on the macrophage that further signals the initiation of phagocytosis. Opsonic receptors detect host-derived proteins bound to target particles. These proteins are known as opsonins and include antibodies, fibronectin, complement, etc. Fc receptors bind to the Fc portion of the IgG or IgA antibodies. Complement receptors bind to activated complement components such as IC3b deposited on the particle. Now, in the second step, we can see engulfment. The macrophage has binded to the opsonin-coated bacteria through opsonin receptors. Upon binding to the particle, phagocytic receptors initiate signaling pathways leading to remodeling of actin cytoskeleton and lipids in the membrane that result in the membrane extending to cover the particle and leads to formation of a cavity inside it containing the bacteria. This is called as phagosome. Next in the animation, we can see the lysosome that contains enzymes responsible for degrading and killing of engulfed bacteria. In the third step, we can see that opsonin detaches from the bacteria and is released out of phagosome, and then phagosome gets surrounded by the lysosomes. The next step is phagolysosomal formation. In this step, the phagosome fuses with lysosomes and becomes a phagolysosome, a potent antimicrobial vacuole in the cytoplasm of the macrophage. This step is also known as phagosome maturation. This completes the fourth step of phagocytosis. In the fifth or the last step of phagocytosis, there is killing of the bacteria in the phagolysosome. Phagolysosome is the fundamental microbicidal organelle equipped with sophisticated mechanisms for degrading microorganisms. It has a highly acidic interior, pH as low as 4.5. The low pH obstructs the normal metabolism of several bacteria. It also contains many hydrolytic enzymes. Some of these enzymes are various cathepsins, proteases, lysozymes, and lipases. The phagolysosome also produces reactive oxygen species ROS and reactive nitrogen species as seen here. You can see superoxide O2 minus, hydrogen peroxide H2O2, hydroxyl radicals OH minus, hypochlorous acid HOCl, and nitric acid radicals NO. The various forms of ROS can damage DNA, proteins, and lipids of microorganisms. Ultimately, the bacteria is killed inside the phagolysosome and eliminated. This ends the discussion for explaining and understanding the process of phagocytosis.